Hello, my name is Armada-Moreira, and the concept of my PhD project was actually quite simple. What if we can make artificial cells that can save neurons from diseases? For that, we looked into astrocytes. These are like the handyman of the brain that allow neurons to function properly. Amongst many functions, they can recapture the synaptically released neurotransmitters, they can detoxify ammonia, and they can regulate oxidative stress by uptaking and neutralizing reactive oxygen species. So when these multifaceted cells aren't working properly, all these tasks will be impacted, and the concentration of extracellular glutamate will rise to toxic levels, culminating in excitotoxicity. This process, which describes the toxic actions of excitatory neurotransmitter, has been linked to Alzheimer's disease, ALS, and other neurological pathologies such as epilepsy and traumatic brain injury. Thus, by tackling astrocytic function and excitotoxicity, we can counteract a very widespread cellular phenomenon that underlies several different pathologies. With all this in mind, we set out to create an astrocyte mimic that can counteract excitotoxicity. First question to be answered: how do you build an artificial cell? For this, we counted with the amazing collaboration of Brigitte Stadler in Denmark to learn all about cell mimicry techniques. And using layer by layer deposition, we were able to create around 20 micron in diameter structures with two key components that can tackle excitotoxicity: platinum nanoparticles that are able to convert H2O2 into water and oxygen and simultaneously convert ammonia into nitric oxide, and glutamate dehydrogenase, an enzyme that can break down glutamate. After building these microreactors, we incubated them with neuronal cells, and we were able to confirm that 1, they are biocompatible, which means that the cells do not die when in contact with them, and 2, that they can interact with the cell membranes. Even when we tried to wash them away, they were still glued to the neuronal membranes. Then we exposed the cells to toxic concentrations of either H2O2 or ammonia and observed how many cells died. When we repeated this process but added to the mix our microreactors, the cell viability increased significantly. But our brain is much more complex than cells being alive or dead. Neurons need to be able to communicate with each other in order for us to be us. So, to investigate whether our microreactors could salvage neuronal communication in the face of excitotoxicity, we plated these neurons, allowed them to mature and form neuronal connections, and then perform extracellular electrophysiology recording, just to check how they communicated. So then we exposed them to H2O2 or ammonia for 24 hours and repeated the same recording. Their response was clearly affected. However, when we added to the mix not only H2O2 or ammonia, but our microreactors, the neuronal responses were similar to those of the baseline. So at the end of this project, we were able to create biocompatible artificial cells inspired by astrocytes that can not only rescue cell viability in the face of excitotoxicity, but also preserve neuronal communication. This project was only a success because of its interdisciplinary nature that mixes molecular neuroscience and biochemistry techniques and approaches with material science and cell mimicry. So I guess my conclusion is, the more you venture out of your field and bridge connections, the more chances you have of making something new and impactful.